Okay, yeah, the title of our presentation today is Monitoring Phytoplankton for Drinking Water Treatment Plants with a focus on cyanobacteria. Yeah, that's what we want to present here, and what we think it is the use of a technique, fluorometry, to determine algae and cyanobacteria. And we focus also on drinking water treatment. So drinking water means that the water that is produced has to have a high quality, very high quality, of course, because it is for drinking purposes. But clearly, water treatment also depends on the demands of what is aimed for, what it is aimed for, the intended use. So we have the treatments, of course, the raw water, the treatments, and there are some standards regarding the quality of parameters, for example, pH, temperature, conductivity, and so on. And to determine the quality, of course, there are sensors or measurements that have to be done to determine the quality more exactly. It means that we have in water processing, we have sensors for pH, temperature, conductivity, or color, turbidity. So what is listed here on the bottom of this screen, and where these sensors are located, that depends on where the water is extracted. It can be, for example, it can be raw water, but it can be also here in a drinking water process treatment at the beginning of the uptake and during the different processes. These are different kinds of water treatment plants here in US America, just as an example. And because of the different parameters, we have to deal also with other parameters, for example, chlorophyll, which comes from phytoplankton, and phycocyanins, which comes from, let's say, blue-green algae. I will explain this a little bit later. And an essential part of the water quality is that the water may be contain also some toxins or which is also unwanted taste and odor compounds. So this can happen that you have some compounds inside the water which is unwanted. And of course, for drinking water treatment, it is very important to omit such substances which can impair the quality of the water. And we have different types of phytoplankton, which can be part of the raw water. And here is a special group are the cyanobacteria. These are not real algae. These are cyanobacteria close to bacteria, but they have the feature that they can make toxins. And what I want to show here that we have different types. I do not want to go to all of these, but it's important to say that even under conditions where there is not a high amount of nutrients inside, some algae, some microalgae, some cyanobacteria can grow even under those circumstances and produce toxins. And here there's a very common cyanobacteria, the microcystis. I will show this a little bit later, and I will come back to this kind of cyanobacteria which can produce such toxins. So, of course, these toxins are unwanted in the uptake of the water, but also during the different processes. So what they have to do in water treatment, they have to omit such cyanobacteria, and of course, other algae, they are also not wanted in the drinking water at the end. So what these phytoplankton toxins can do is they can cause hepatotoxicity or neurotoxicity, respiratory paralysis, and at the end also cell death. And I have here some or two compounds. This is on the upper part, it is the microcystin made by this microcystis I've shown in the slide before, which is a phosphatase inhibitor. This can cause the liver disease, and it is a cyclic oligopeptides, and it's working well in a longer time, in longer period on human health, but also on um, on, uh, dogs and cats and other animals when they drink this water. And some other toxins like this one here. This is the anatoxin, is a very fast deadly factor. It's an alkaloid which reacts very fast on ion channels, and this can cause immediate death of organism. So what I want to say is these toxins can come with this raw water and has to be omitted during the process. So it's important to know when the water is contaminated with such cyanobacteria. Here in the background, you see an algae bloom of such an blue-green algae, the cyanobacteria called oscillatoria. Toby will show more about this later. Unfortunately, these cyanobacteria are not limited to a certain period. They can come over a very long time of the year. And you see here also microcystis, this blue-green algae showed at the beginning, but different species. And when you look over the distribution in time, they can vary over the year. And even in wintertime, when the sun is not that bright and the algae will not grow so good, you can see that these cyanobacteria can occur and can contaminate the water. So it is that means that it's important to monitor during all the year, even in winter times, when the photosynthesis of the cyanobacteria is rather low. Yeah, when you look at this picture here, on the right side, you see a reservoir, a picture which I made in Poland many years ago. And you see a bloom. And what they are doing in this reservoir, they try to monitor the cyanobacteria because they want to get only water within a rather good quality. Here, the wind drives the cyanobacteria to one end of the reservoir, and here these algae cells are broken and release the internal pigments, but also the toxins. What people are doing then now, they want to detect these algae or cyanobacteria in a very early stage and can control the risk of harmful algae emitted especially by the cyanobacteria. And for this, there are some parameters they can determine. This is, for example, take samples and measure the cell counts, or determine the biomass, fresh mass or dried biomass. They can also determine the biovolume. Sorry. And an important factor, which because it's rather easy to determine, is to determine the chlorophyll content or the phycocyanin content. These are both pigments which are inside of the algae cells and the cyanobacteria. And sometimes they use also other key pigments which are inside of the cells. I will come to this a little bit later. That needs a more sophisticated type of measurements. There are not clear rules for this, what is allowed in drinking water, but here there is a recommendation by the World Health Organization. You see it here on this screen. They measure either cells per milliliter, or they measure chlorophyll. Here they give the levels which are allowed or which are alarm levels for drinking water. This is just to omit the content of the toxin inside of the cells. Why can we use chlorophyll as a proxy for the toxin? Well, it is said that let's say around about 50% of the cyanobacteria can make these toxins. So chlorophyll of cyanobacteria is let's say an indicator for the toxin, and I have a thumb number which is around about 1/10 of the chlorophyll content is the content of toxin. So if we have, let's say, 10 microgram per liter of the cyanobacteria, then we can expect that we have 1 microgram of microcystin inside of this water as a toxin. This is just a rough number and it can vary, but just an idea how much it is. So to determine these chlorophyll content, we have different types of measurements. A very common way is to extract the pigments in organic solvent and determine it at different wavelengths in the spectrophotometer. This gives you an information about the chlorophyll content, but not the information what kind of algae it is inside. So more helpful is the high pressure liquid chromatography, where we can also extract the different pigments, but separate them on a column and determine the quantity afterwards in a special way of photometry or fluorometry. That means that is also a kind of spectrophotometer, but we have now the pigments separated and we can sort it. Another way, what we want to show here in application, is the fluorometry. Here we do not need to extract it. It is an in vivo measurement, and it gives you an information about the algae classes or the cyanobacteria more in detail. And the advantage is here also that we have a rather sensitive, because the fluorometry is by a factor 10 to 100 more sensitive than the spectrophotometry. So how it works is quite simple. We have the light. It can be artificial light or sunlight, and we illuminate the algae cells, and we have here the so-called peripheral antenna, which are a composition of different pigments which lead the energy to the internal cells. And some of this energy is fluorescence, so it is light which is emitted. You see it here on the left side. This is a microscopical picture of green algae, and when you illuminate them, you find some fluorescence, the red light here, which is emitted. But of course, for the cell, it's important that they can make energy, like starch, for example, that is used for reproduction of the cells. So this can be seen as a waste, but it is something which can be used for the determination of the chlorophyll content of cyanobacteria and microalgae. Fortunately, the different algae and cyanobacteria, they have these so-called accessory pigments, and the accessory pigments, they can affect the chlorophyll efflorescence, this red light which is emitted, and so it depends on the accessory pigments what kind of algae we can see and distinguish with the fluorometry. For example, we have here the blue-green algae, or the cyanobacteria. They have the phycocyanin, which I showed in the beginning, and this pigment affects also the fluorometry. And with the different accessory pigments, we can sort the different algae classes and cyanobacteria. So what we can measure here, up to 5 different algae classes with this kind of fluorometry if we use different light sources. And in this picture, I would like to show you how these different algae and cyanobacteria depend on the excitation wavelengths. So what we use in fluorometry, we use different wavelengths to excite the pigments together with the accessory pigments. And what you see here is, for example, let's take this curve. If we excite at different wavelengths the cyanobacteria, we get the emission or the fluorescence, which is shown here as a signal. And another group, for example, the green algae or the chlorophytes, that is this curve that shows you what is the amount of fluorescence emitted when you illuminate with these wavelengths. And with this knowledge, which we got from a library of algae, we can determine the amount of different algae class and cyanobacteria in a mixture in vivo measurements in raw water. And in raw water or raw water extraction, we use, let's say, a continuous monitoring, and you have here a diagram of the Algae online analyzer. You see here a flow-through tube, and this is the flow to this through this tube. And from light side here, from the left side, you see different wavelengths, LEDs, which act as in high frequency and illuminate the algae cells which flow through this tube. And if they got excited, then they will send out the fluorescence, and the fluorescence is measured with a photomultiplier. And of course, we have a filter here to remove the exciting light, and we only measure the fluorescence. And this goes rather fast. That means we can measure in a very short time. And after that, we have, of course, a software which can determine the amount of different algae in a mixture. And this is the instrument where we have the sensor here on the left side, which show with the tube, the intake and the outlet. And then we have the signal that goes to the PC here, and internally is calculated what is the amount of the different algae in a time course. And what you see here is the are the diatoms here, and you see the total, sorry, you see the total chlorophyll content and the green algae. So you have a time course, uninterrupted measurements of these algae and cyanobacteria that flow through this sensor. And for improvement of the measurement, we have also a cleaning device included. That means this removes the biofilm inside of the tube, so that you have continuous measurements at 24 hours, 7 days, uninterrupted. And for better analysis, you can also add thresholds for this that will release an alarm if a certain chlorophyll content is superseded, for example, for the unwanted cyanobacteria. So I think this is all for the intro of the chlorophyll measurement with the Algae online analyzer. And now I want to pass this over to Tobias, because he wants to come to the second part. So.
Yes, good afternoon again from my side, and I will continue with more applications. Yeah, and if you, I don't know, if you want to see me in live, I come to Australia 20th of August, July, and after exhibition we have maybe some workshops with Thermo Fisher again. I want to tell you some specs, what is very important at this time. We calibrate with real algae, living algae. We grow them in our laboratory. Always will use them for calibrate. And for example, you see here some blue-green algae, green algae, brown algae, and so on. And we grow them, and we try to make a good as possible calibration of the instrument. Here you see that we really grow them, and we get sometimes also some new algae, and to have a high standard of calibration possible. I will tell you a little bit more about the application. Here, Detlef is sitting more close to the Danish border. I'm sitting here, and Babak sits close to Kiel, far north of Germany. And there's a very big drinking reservoir here in North Rhine-Westphalia, and they have 12.6 million inhabitants, 130 liter a day. They have 2.2 million cubic meter a day in this dam, and they give drinking water to this area here. The Seckenbach Talsperre or Seckenbach drinking water reservoir is 20 hectares, 2.8 million cubic meter, and the depth of in the middle is 36 meter. And what they found, an algae problem. There is also a recreation area around. You can just walk around, and the drinking and dog was drinking in the water and get death after a while. And then they asked at the drinking water production area, what's going on with your reservoir? And then they checked and found Panthotrix rubescens. It's a Burgunder blue Burgunder algae, and you see that is a network, and the Panthotrix rubescens has a high toxicity in this water body. Afterwards, they installed an Algae online analyzer. You see it here as a typical installation. You have pH and turbidity as standard parameters, and they have also used Algae online analyzer at the waterworks and measure the raw water and filtrated water, and you see the different content of the algae. They have also a valve system, and in the first step they measure the raw water, which is on the right hand side of this, and after the ultrafiltration they measure the second step, and then they can see if it happens. I spoke with the operator of this instrument, and if there is a high content of algae, they integrate also a cold filter, so that they just use the cold filter to be sure that there are no algae in again, and they can use this instrument to reduce the cost of this measure of this waterworks, because they only use the cold filter if there is a high content of algae, and so they can get some cost reduction. Just to give information about the cell counting and our measurement, the green line is our measurement from the Algae online analyzer, and they had made also some with the red dots, some measurements with the cell counting with the microscope, and I can see it works quite okay. And the rough of this of our line is because the Panthotrix rubescens, sometimes there are a lot of algae going through, and sometimes not that much algae going through. They also use a laboratory instrument to get some information at the intakes, and they take samples on the rivers who get into this lake to get an idea on where the algae or the cyanobacteria comes, and they make also profile measurements on different depths. I will show later on. Yes, and they use a fluoroprobe to make depth profiles to see where is a high peak of the cyanobacteria. I come to another spot. It's a treatment plant at Clear Lake. It's in California, US, and then there's a Clear Lake at Buckingham Park. They have a treatment plant. It's a very popular recreation and fishing spot, and they have sometimes really big problems with algae. We'll show you later on. Yeah, they have an intake pipe of 10 meter steps, sedimentation and fluctuation, typical treatment steps. then pressure filter, and they monitor with our lab instrument the content on the different treatment steps. Some samples as a case study of approximately 3 years algae in Buckingham Park. Thanks to Will, who makes all these measurements, and you see here the distribution of the different of 1 year. You see 3 years. It's May 2019 until February March 2022, and yes, that's a very long long data track. If you just look for this area, you see what we are measuring. We measure green algae, diatoms, blue-green algae, plantotic rubescens as a red type of cyanobacteria, and yellow substances. And if you see here, we have a strong increase of all algae classes. There's the total chlorophyll green and diatoms, and you see a very strong increase over the years, about 3 years of the whole algae content. And if you see typical on the northern hemisphere that you have in spring diatoms and green algae dominated. That's in Australia would be on the opposite side, and then you have typically the cyanobacteria in the more autumn area of the year. So it starts with diatoms and green algae, and then afterwards the blue-green algae start with that. If you look for the cyanobacteria, the variability of the cyanobacteria, it has an increase of 4 times higher within the years, and that's a dramatic increase of the toxic compounds in this lake. What is potential toxic compounds in this instrument? As I already told you, I see all the time in autumn you have the high peak of cyanobacteria in this lake. It's also an interesting result that the duration of the cyano blooms gets longer and longer. At the beginning, it was only 3 months, then 4 months, and now it's more or less over the whole year some content of cyanobacteria, and that's was an interesting result. Some installations. Here you have a Kentucky water treatment plant. They have a Algae online analyzer. Here on the left-hand side, the sensor is inside, and they measure 4 different treatment steps in this instrument, and they just get 24/7 with the results. And they want this. I was there for the first installation, and now they will install it to the operating panel to get the information about this algae. Another installation is in Toronto water treatment plant. You see the Algae online analyzer on the left, on this side, or on this side. That's a typical installation. They have here 4 valves and 4 intakes, and they measure 5 minutes the raw water, 5 minutes the first treatment step, and so on. And with this valve system, they can change the different intakes. On the right-hand side, it's only 2, but it's the same. They have 2 treatment plants, and one is with 4 valves, and the other is with 2 valves. The typical application is on a ferry box system. You see here that's a flow-through instrument, and you measure PCO2 or dissolved oxygen and other parameters with this instrument. And here you see they integrate our Algae online analyzer sensor to get the content of the different algae, and that's a typical application to have an automatic system for different parameters. And here in Middle East, we have an installation. We have also here BBE Algae online analyzer. They have also a fish toximeter there. And that's also a typical installation to get an idea about the water body. maybe you heard of it already in Lake Erie, that is in the north of, it's one of the Great Lakes in North America, North America. And you see here Toledo, and you see a satellite picture of the cyanobacteria bloom here. It's 2011. And then in August, the 2014, they awoke to urgent warning, not drinking the water. There were toxins from algae bloom growing in Lake Erie, and now they have the drinking, the bottled water was sold out there, and it was a very harmful bloom, and it is all the time the problem if the waterworks has such a problems and do not inform the free people, then they trust it. Now 2014 to 2025, now they start trusting the waterworks again. But if before it was all the time, what are they really working good or not? That's the intake of the waterworks is outside of the lake, and you see the blue-green or green or algae bloom in front of it. Just the information about the depth profiling. We have a Seepark Lake in south of Germany. You see here is a fluoroprobe and an algae torch, and they measure depth profiling of this instrument. And with this instrument, and you see at 10-meter depth, you have the oxygen goes from more or less saturated situation to nearly zero. And you see the different algae, the temperature of our instrument, total green algae, diatoms, Plateautrix rubescens, blue-green algae, and yellow substances. And if you see only the cyanobacteria, there's a big peak at the thermocline, and that's often where the intake of the waterworks going in. And you see that the Plateautrix rubescens and the blue algae has a big peak. And also the yellow substances has a peak here. That is the situation where the cyanobacteria like to live because they don't need that much light. But there are a lot of nutrients in this area. And the peak is sometimes a problem if you are the intake of the waterworks are more or less at 9, 10, 12, 15 meters. And sometimes it's a problem that the cyano bloom is in this depth very, very high, and you can get easily the cyanobacteria in the waterworks, and that is the problem to get to be aware on this. And with the Algae online analyzer, if you measure the intake of the waterworks, you get the content, what is really going in your water body. Just the last slide is a Fullback reservoir. It's a dam there, and they have a drinking water producing area there. And they use also an Algae online analyzer, and they made also some measurement, and they aware that there's a drinking water area. And if you see here also the different algae distribution, if you use only satellite pictures, you don't see the algae distribution in the depths. And that is also important for be sure what you are measuring. And the temperature profile on this situation. And we have some area where the content of the different algae are very high. Just a seasonal change, and if they in March, if they start growing, the concentration of chlorophyll A and cyanobacteria is very high on the deep areas, and then there is a mixture in the lake, and then the cyanobacteria go also Plateautrix rubescens here on the surface area, and sometimes they say that there's a dramatic increase of the bloom, but it is more or less a mixture of the water body and the toxins which are all the cyanobacteria which are already there go up to the surface area. Just some information about our flow meter. We have a lab analyzer. You can just take a sample, 25 milliliters, put it inside, and within a few minutes you get results. Then you have this Algae online analyzer, which is a typical instrument on waterworks because it's easy operating. You have a cleaning system inside. You have algae torch, bento torch for handheld instruments, a fluoroprobe for depth profiling, and we have a new Ulrika probe from US. We combined it with our new fluoroprobe, and you can also measure dissolved oxygen conductivity and pH. We have also some toximeters, but that is not part in this. Algae, Daphnia, and fish toximeter. And thank you